Okay, today's lesson, we're going to look at chi-square test. Now, if you remember, chi-square test, right, is one of the statistical tests that I've introduced to you when I introduced paper 5. Remember, in paper 5, there's a section where you need to do statistical test or statistical analysis, all right? And all those that you have learned includes range, interquartile range, standard deviation, standard error, error bar, as well as t-test. Okay, so chi-square test is another statistical test, which is quite interesting, very similar to t-test. So if you're familiar with t-test, right, this one is actually very, very easy because the principle behind the test is similar to the t-test, okay? Okay, so for chi-square test, now we have just learned the genetic diagrams, right? So when you use a genetic diagram to work out a cross, right? So you do all the calculations, the genetic diagram, then you get your offspring's phenotypic ratio, for example, 1-to-1 ratio. Okay, so this one is 1-to-1 ratio. So for example, you've done a calculation, it's 1-to-1 ratio. Okay, so this is the, then you go and carry out the experiment. So instead of doing calculation, you go and do the cross, okay? Usually for genetic crosses in the lab, right? What we'll do is we will cross the fruit flies. You know, the tiny little fruit flies, we'll cross them because they will, their life cycle is very, very short. So we can see they will lay eggs, and then after the eggs will hatch, and we'll see the offsprings in a very short time. Okay, so let's say if you do the cross in the lab, and then the offspring phenotypic ratio that you will get, you'll be expect that is called the actual results, your actual results from the experiment, which is also called the observed results, because you can see observed result represented by symbol O. You expect it to be quite identical or close to the prediction. Prediction is what you have calculated using genetic diagram. So that is called the expected results, or E. Okay. So you expect it to be close, but you will never expect it to be exactly identical. All right. So your observed results that you get here, you expect it to be close to your expected result 1-to-1, but it will never be identical. Why is not identical? Because every experiment, there will be experimental error. Okay, for example, the eggs did not hatch, or the flies did not mate, or the flies died, okay, before laying eggs, or the larva died inside the food that you provide to them, okay, the jelly-like fruit. So a lot of reasons because of that. These are all the experimental errors. So your observed ratio, phenotypic ratio, will not be exactly the same experimental ratio because of the experimental error. But you expect them to be very close. So how close? So if a person gets 10-to-13, can this be considered as 1-to-1? Can, right? Because it's very, very close to 1-to-1. How about 10-to-14, 10-to-15? So until when we can say that, okay, now your ratio is too far away from 1-to-1, we cannot say that it's 1-to-1 ratio already. So to what extent that to what extent is the cut-off point? Okay, hang on, is it charging? Okay, all right. I hope my iPad is charging, hopefully. I hope it's charging. Okay, let me check whether it's charging or not. Yeah, it's charging, but very slow. Okay, hang on. Hang on, I go and get a... So we want to find out how far should the O and E be so that the O is no longer equals to E. So we want to know what is the cutoff point that we say that, okay, now your observed ratio is so much different from 1-to-1 that I would say that your observed ratio cannot be considered as 1-to-1 anymore. Okay, so we want to find out where is the cutoff point. So in order to find out, we have to do chi-square test, right? So chi-square test is actually a goodness of fit test. Chi-square means chi-square, okay? It's a goodness of fit test. That means you want to find out, it's a statistical method or statistical test to assess if the O and the E ratio are significantly different or not. Okay, that's why it's called goodness of fit test, because you want to know whether O fits with E well or not. Okay, whether O and E, they fit each other well or not. So goodness of fit test. So to assess whether O and E are significantly different or not. Okay, so this is a formula for chi-square test, and the formula will be given to you in exam. But you need to know that in order to calculate chi-square value, you need O and you need E value. Okay, so this formula chi-square is, so this is chi-square, this is E is expected value that you calculated from a genetic diagram, and O is your observed value that you observed from your experimental result, and this is sum of, okay? So let's say if I get your whole class to do the genetic crosses in the lab, and I ask you to calculate your chi-square value, and I collect the whole class chi-square value, I will be able to plot a graph like this. So this is a chi-square distribution graph, okay? Chi-square distribution graph, whereby the x-axis is the chi-square value, okay? And the y-axis is actually the frequency, okay? The frequency or the percentage, okay, of your entire class, right? So this is a chi-square value. So we're going to study this chi-square value. Just remember the formula for chi-square. So I'm going to bring you to another region where we can actually do this sharing. Okay. Now, remember your chi-square. Okay. Chi-square is equals to summation of O minus E divided by E. Okay. Square. Okay. O minus E square. Okay, so this is the formula, all right? So I want to look at this chi-square distribution. Let's say I got it from the whole class, okay? Now, this is actually the chi-square value. So looking at this point here, when chi-square is equals to 0, if you look at the equation, right? When chi-square is equals to 0, what is the probability of O equals to E? Okay. What is the probability of O equals to E? Let me see. Jocelyn Chang. Please give me your answer. The rest of you, can you please type your answers in the chat? So what is the property of O equals to E when chi-square is equals to 0? Looking at the equation. Jocelyn? Please unmute yourself if possible.
Yeah, I unmute myself.
Yeah. The rest of you, can you please type your answers in the chat so you can have some interactions here instead of you sitting down there. Okay, you can respond, you can write your answers so we can see.
Can you repeat the question again?
Okay, look at the graph. I'm pointing at the x-axis, right, when the chi-square value is equal to 0, isn't it? Correct?
Yeah.
Okay, so if you look at the equation for chi-square, look at the equation. So when is it chi-square is equals to 0 based on the equation?
When O equals to E.
Very good. When O equals to E, because O minus E becomes 0, isn't it? So your chi-square is 0, right? Okay, that's correct. So chi-square can only be 0 when O equals to E, right? So what is the property of O equals to E when chi-square equals 0? You say O equals to E, right?
Yeah.
So if O equals to E, property is 100%, isn't it?
Yeah.
Correct? The property of O equals to E, because you know, you already said O equals to E, that means property of O equals to E is 100%, right? Correct?
Yeah.
Okay, correct? Huh? So this is 100%. Or another way to write 100% is equals to 1. Okay, if you don't put the percent, it means it's equals to 1. Okay, can follow?
Yeah.
Now, so we're going to look at the x-axis now. So as we move to the right side of the x-axis, this way you can see that the value of the chi-square value actually increases. So chi-square value actually increases, correct? As you move to the right side of the graph. So when chi-square value increases, what is the probability of O equals to E? Does it increase or decrease or remain as 100%? Let's call another person. Hey, the rest of you, can you please type your answer in the chat, because I want some interaction with all of you. That would be very, very nice. Hua Hui Tian?
Yes.
Okay, good. What is property of O equals to E as the chi-square value increases?
Decreases.
Very good, decreases. Okay, very good, because when chi-square value increases, it increases because this value increases, isn't it? And this value can only increase when O and E becomes more and more different, right? More and more different. So the difference between O and E becomes more and more different, so chi-square value increases. Okay, so based on this formula, the property of O equals to E has decreases, right? So you can see that in this chi-square distribution, it's actually telling you that when you calculate, after you've done experiment, you calculate your chi-square value for your experimental results. Now, you can check the location of a chi-square value. If it's located exactly at chi-square equals to 0, that means your property of O is equals to E is exactly equals to 1. Okay, O is equals to E. And the further away you are from 0, okay, along this graph, that means the more different is your O compared to the E ratio. Okay, remember, so the more different it is. So we are actually trying to look at what is the cut-off point, so that we say that, okay, your O ratio is so different from E that we can no longer accept that O equals to E. So we want the cut-off point, right? We can allow experimental error, but we cannot allow too extreme errors. You have to cut off those values that have got extreme error one. So based on this graph that I get from your whole class result and calculation, I will determine. So in biology, usually the cut-off point is the final 5%. So these are all the results, 100% of you, they are very close to the original value, okay? And then these are the 5% that the results is very, very far away, the 5% end, okay, of the whole group of you. So therefore, the final 5% is actually this region here. Okay, because these are the result where the O is so different from E, they are at the 5% end. So whenever any chi-square value that falls within this range here, we will say that, so we want to find out, right? So whichever that falls within this range here, we will say that O is significantly different from E already. Okay, and whichever your chi-square value that falls here, we say that there is no significant difference between O and E. So this is where we are using chi-square to find out the cut-off point. So the cut-off point in biology is usually the final 5%. So therefore, at this value here, so this is called the critical, okay, can I type here? Okay, here, yeah, I cannot type. Critical chi-square value. Now, critical chi-square value is actually the chi-square value when property of O equals to E is exactly 5% or 0.05. Okay? So at this point here, property of O equals to E at this point here, okay, is exactly 0.05. Property of O equals to E. So as you move to the right side from this point, okay, as you move to the right side from this point, the property of O equals to E becomes actually less than 0.05. Okay, so therefore, after you've calculated your chi-square value, you just have to check where is the location of your chi-square value. If your chi-square value is located at this shaded region, then you know that O is significantly different from E. Okay, and then if your chi-square value is located at the 95% region here, okay, then you know that there is no significant difference between your O and E. Is still acceptable. There's the differences is because of random experimental error. Okay, whereas the differences here is no longer random. Okay, it's two different that we cannot say that it's just random error, experimental error. Okay, so this is a principle behind the chi-square test. All right. So so now we're going to move on to do an actual chi-square test. All right. So here, any chi-square located here is considered as the acceptable difference, and those located at the shaded region of the chi-square distribution graph is the rejected differences, and the one, the chi-square value where probability of O equals to E 0.05 is called the critical value. Okay, critical value. So in Cambridge A-levels, you are expected to use chi-square test to analyze results from either genetic or breeding experiments or ecological sampling. So only these two, because these two deal with organisms. Okay, so they deal with organisms. Either you're doing genetic diagrams and then you do genetic experiments, or you go to the environment to count the number of organisms. Okay, so they're all dealing with organisms. So therefore, chi-square test is only done for data that are discrete. Discrete means number of individuals, right? So you don't have decimal place on because you're dealing with organisms. Okay, so discrete units. So your data are always discrete unit 1, 2, 3, 4, 5. Then your data is categoric. Categoric means, for example, if you genetic diagram, you say, oh, eye color is brown to blue. So you got two categories. Cat coat color has got 3 colors, Burmese, Siamese, and Tonkinese. So you got 3 categories. And ecological sampling, let's say you say, okay, I want to find out the number of snails on top, middle, and bottom of the hill. So your category is top, middle, and bottom of the hill. So you got 3 categories. Okay. So the nature of the data is discrete and categoric for chi-square test. So the first thing you need to do for chi-square test, you need to set up null hypothesis, because when you want to test something, you must have got some statement for you to test. So your statement is always null hypothesis. Null means 0, okay, H0, H subscript 0. So null hypothesis statement is always there is no significant difference between O and E. So the difference is 0, right? So H0. So there is no significant difference between O and E. That means any differences between O and E is due to chance or is within the acceptable range. Okay, so this is the first statement you need to do. Then second, you have to calculate the chi-square value, okay, using the data that you have. So I'm going to move you on with the calculation now. I don't want to see all the notes. We have to do the calculation, then you understand, okay, because this is maths. Okay, so let's say, for example, in an experiment, the expected ratio is 1:1, and the observed ratio is 110 dominant and 90 is recessive. So now you're asked to do the chi-square test to find out. Okay, to find out the goodness of fit between O and E. So step number 1, let null hypothesis be there is no significant difference between O and E. So only when you do a statistical test, you can use the word significant. difference or not. If you don't do statistical tests, you cannot use the word significant, okay, because you're not sure one. But when you do the test, then you're quite sure with a certain degrees of confidence, then you can use the word significant, okay? Okay, so now you calculate the chi-square values, step number 2. So this is a table that sometimes they give you an exam for you to do the calculation of chi-square values step by step, okay? So observed ratio for dominant 110, recessive 90, all together population 200, okay? Expected ratio is 1:1. So for 1:1, if your total is 200, then your 1:1 should be 100 and 100. Okay, so you need to write 100 and 100. Then after that, you do the calculation step by step. So what is O minus E squared, and O minus E squared divided by E? Okay, so do the calculation, and then after that, add up the summation for the 2 categories. So this is equals to 2. I think you need to fill in your notes, huh? So now you've calculated your chi-square value is equals to 2, all right? Are you done with the calculation? This is very straightforward, okay? Okay, please fill in the blanks in your notes. Do calculation yourself, don't copy mine, okay? Then you can verify. Okay. Then the next step is you need to calculate DF. DF stands for degrees of freedom, okay? Degrees of freedom. Okay, the degrees of freedom formula is V is equals to C minus 1, whereby C is the number of category or the number of classes. So here you got 2 categories, dominant versus recessive. So therefore, your degrees of freedom is 2 minus 1. Okay, so it's equals to 1. All right? So this is degrees of freedom. Okay, I think there's something there for you to work on for degrees of freedom here, this slide. Okay, this example of how to calculate degrees of freedom. So for here, you got 2 categories, degrees of freedom is 2 minus 1. Okay, this one you got 3 categories, degrees of freedom is 3 minus 1. And this is 4 categories, so degrees of freedom is 4 minus 1. Okay? All right. So let's go to the next one. Okay. So the next step, after you calculate chi-squared value and the degrees of freedom, now you can go to the chi-square table to find the property of the chi-square value. What is the property of this chi-square value? Okay? So this is a table of distribution of chi-square, and this table actually represents the graph that I've shown to you just now. Remember the graph? So the chi-square value is located in the body of the table. So all these here are the chi-square value. Okay, so you can see that this chi-square value starts from very small value and it starts to increase. So therefore 0 is here. So this is actually the chi-square value, okay? So all these values in the body of the table, they're chi-square value, okay, which actually represent the chi-square distribution curve. So now you need to find out where is the location of your chi-square value for your experiment. So just now we have calculated that the degrees of freedom is 1. Wait, 1, okay, so it's here. So this column represents degrees of freedom. So you have to look for the correct degrees of freedom. So it's 1, okay? And then go and find out the location of your chi-square value. So your chi-square value just now calculated is actually 2. Okay, so let's look at where the number 2 is located. So 2 is located here, right? Between 1.640 and 2.706. So between these 2 numbers, okay? So therefore, after find out, you have to write. Okay, you find out here already, you see what is the probability of this chi-square value, okay? Now you see here, this is actually probability, 0.995, and this is 0.05. This is 0.005. So this is actually showing the probability of O equals to E based on the chi-square value. Okay, so when a chi-square is located here, okay, located here, then what is the probability of O equals to E is between these two, right? So since probability of your chi-square value is 2, degrees of freedom of 1 is equals to 1, is located here, which is actually between 0.10 to 0.20. So this is the first statement you need to write. So the property that you find out from the table is between 0.10 to 0.20, okay? And then you compare this property with the cut-off point probability. The cut-off point probability is 0.05, okay, the cut-off point, okay, which is this one here. So that's why we divide the entire table into 2 sections. Remember, this section is actually 95% of the data, and this section is the final 5% of data. So this is the rejection zone. This is the acceptable zone. Okay, so your probability of a chi-square value is between 0.10 and 0.20, and then this is actually less than 0.05 probability. So it's less than, sorry, it's more than 0.05. So it is located at a zone where null hypothesis is accepted, right? So null hypothesis is accepted whenever chi-square value is located in this area. So then you need to do your conclusion already. So what do you mean by null hypothesis is being accepted? That means you have 95% confidence that there is no significant difference between O and E. So which means O is equals to E, okay? So this is the conclusion statement that you need to make. So you have 95% confidence that there's no significant difference between O and E. That means O is equals to E. And any difference that you can see is by chance, right? Now, why is it you need to write 95% confidence? Is because when you do the test, you have decided any data that are located at the 5% end, okay, I will reject it. And sometimes you may reject wrongly. So therefore, when you make this decision, how confident are you that your result is correct? Since you got a 5% chances of rejecting wrongly, that means your confidence level is 95%. Okay, because when you reject the 5%, you may reject wrongly. So it could be a mistake. So how many percent will not be a mistake? 95%. So therefore, how confident are you with your data that is correct? 95% confidence. Okay? Okay, so this is the first way that you find out for your chi-square value, what is the probability, and then you can do your conclusion. Okay? Now, the second way in exam that you may find out what is the probability is they don't give you the table. Okay? They will not give you the table. What they give you is the critical value only. The critical chi-square value. That means they'll tell you that, okay, the critical chi-square value for degrees of freedom 1 is 3.841. Degrees of freedom 2 is 5.991. And then degrees of freedom 3 is 7.815. So they'll give you this data. Okay, then you have to decide which degrees of freedom you read lah. Okay, so that means to say that now the critical value, the chi-square critical value, right, it's a chi-square value when the probability of O equals to E is exactly equals to 0.05. This is already the starting point of rejection area. Okay, so this is an area whereby this is the area, it's a rejection zone area, remember, whereby O is significantly different from E already. Okay, so now, just now your calculated chi-square value is 2, right? So 2 is actually located here. So therefore, the value 2 is actually less than 3.841. So since it's less than, so your chi-square value is actually less than the critical value, okay, based on your experiment. So therefore, your probability is actually more than 0.05 because your probability is here. Okay, this is more than 0.05. So when the probability is more than 0.05, O has no significant difference from E. Okay, now what happens if your chi-square value is more than critical value? That means your chi-square value is located on the right side of your critical value in the graph. Right side means the rejection zone already, okay? So the property is already less than 0.05. So therefore, O is significantly different from E. So this is the conclusion that you need to make, okay? It's very, very easy. Cut-off point is 0.05 and above, okay? So what is the reason if O equals to E? So you've done the genetic diagram, right, to calculate the expected ratio. Then when you do your experiment, you find, hey, your observed ratio is so different from E. So what could have gone wrong? Okay, these are some possible reasons. Number one, it occurred by chance because the sample number is too small. Say, for example, you expect your F2 phenotypic ratio to be 9,331, but your total number of offspring, let's say you only got 5 offsprings. So if you got 5 offsprings, right, how can you get a ratio of 9,331, right? 9,331 means at least it's got 16 of them or multiples of it, right? So when the sample number is very small, the chances of you to get close to your expected ratio is actually very small, okay? First reason. Second reason is differential motility rate. Differential motility rate, motility means death. It means that, okay, even though your calculated calculated ratio or expected ratio is 9,331, but let's say the ratio you get is maybe 7101, okay, say for example 7101, okay, 01. So how come it's not 9331? How come it's 7101? Okay, so it could be because of differential motility rate. Okay, it means to say that whichever offspring that get this type of genotype for this phenotype, they may not survive as well as the rest of the phenotype. They may need extra nutrients. They may need a lot of certain nutrients like potassium, magnesium that they cannot get naturally, so therefore they cannot survive, they die. Say for example. So this is called differential motility rate for different genotypes. Therefore, certain phenotypes you cannot see, okay, because they die. Third reason is other reasons that affect the final phenotype. It could be what you have learned. You don't get 9331 because of genetic interactions, epistasis, or autosomal linkage. So you thought that the 2 genes coding for the 2 different characteristics, they are unlinked genes, but actually they are linked genes. So, okay, so they behave like a monohybrid cross instead of dihybrid crosses. Okay, sex linkage. So if it's sex link, it has to do with you have to, when you do the genetic diagram, you have to include the X chromosome and the Y chromosome. But when you've done a calculation, if not included, of course the ratio is different. Okay, crossing over. So sometimes we, when we do genetic diagram, we never consider crossing over. So crossing over may occur for autosomal linkage that will give rise to recombinant classes. Okay, so this is a past year question, an example of chi-square test for ecological sampling. Okay, so I want to use this example to show you how to put the expected ratio for ecological sampling. Because ecological sampling, you don't do genetic diagram. So how do you get the ratio for E? Okay, so there is a grassland being grazed by goat. It was compared with grassland ungrazed for varying amounts of time. So moths were trapped and the numbers counted, and then the moths were released. The effect of grazing on a species of rare moth was investigated. So that means they want to see, so there's this land that's not grazed and they count the number of moths. Then there's this land that they start grazing by the goats, okay? And how does it affect the moth, right? So you're asked to do the chi-square test to test the significance of the results obtained, okay? So that means they want to find out grazing versus not grazing, will it affect the population size of the species of moth, okay? So the null hypothesis here is grazing has no effect on the size of moth population, okay? Because why? Because O equals to E. your expected ratio here is actually 1 to 1 to 1. Okay, for ecological sampling, it's always 1 to 1 or 1 to 1 to 1. Okay, what they're trying to show is that whether not grazed, so category number 1 is not grazed, category number 2 maybe is grazed for 1 year, category number 3 is grazed for, let's say, 2 years. Okay, so you've got 3 categories. So whether grazed or not grazed will not affect the moth population. So this is always your E. Okay, your ratio is always 1 to 1 to 1. And then you want to go to the field and then you do the sampling. After that, if your chi-square actually shows there's a significant difference between O and E, okay, then it will tell you that, okay, actually grazing has effect on the number of moths, because you start by testing whether grazing has the effect on the moth population size and not by saying that there's no effect. That's why it's 1 to 1 to 1, whether grazed or not grazed, grazed for 1 year or 2 years. Okay, ecological sampling always 1 to 1 to 1. Okay, so let's look at the data given here. So they actually give a table to students for them to fill in. So O is given, but E you have to decide. Since it's 114, right? If you decide what is E, that means it must be 1 to 1 ratio. So 1 to 1 means 57. Both of them are 57. Then you do the calculation and find out your chi-square value. Okay? So please complete this in your PowerPoint notes. Okay, this is the answers given there. Okay. Why is it? Oh, wait. I see wrongly. So sorry. This is not the total. Okay. These are the 3 different categories. Okay. You add up these 3 is the total. So from the total you divide it by 3. So 824. So this is 240. 240. Okay. So each category you've got 80. So altogether your total sample size is 240. Okay. So after calculation, then you find out what is your chi-square value by add up these 3, because the summation of chi-square value is summation of this one. Okay, so this is actually your chi-square value, not this one. Okay, 39.9. Okay, so after calculating chi-square value, your degrees of freedom, you got 3 categories. So the degrees of freedom is equal to 3 minus 1 equals to 2. So degrees of freedom is 2. Chi-square value is 39.9. Okay, so then they give you a table here. So degrees of freedom is 2, and your chi-square value is 39.9. So look for the location of 39.9. So 39.9 is located here, isn't it? Okay. So probability of chi-square value equals to 39.9, degrees of freedom equals to 2, is actually less than 0.001. So this is 0.01. So it's located here means less than 0.01. And then the probability that you compare with is 0.05. Therefore, probability is actually less than 0.05. So therefore, you got 95% confidence that there's a significant difference between O and E. So you need to write what is your O, what is your E. Okay. So there's a significant difference between grazing versus non-grazing on the population size of the moth. So you need to write in full, okay, when there's a case study given to you. All right. Okay, I like this question here. Okay, I like this question here. So after you have gotten your result, they ask you to explain the significance of these results and suggest the consequences to this species of moth if grazing land is increased. You see, these are the questions given, right? So up to this state here, they ask you for probability. So you give the probability is less than 0.01. You allow reference to it is actually less than 0.05. So you have not done your conclusion yet in the question. So question part 3 is for you to do your conclusion. So what is the significance of results? So do your conclusion, and from conclusion, suggest the consequences if grazing continue. Okay, so 3 marks for conclusion. So first thing you have to do is that, okay, there is 95% confidence. You need to write up. There's a 95% confidence that there's significant difference between grazing and not grazing on the moth population. Okay, and you can also say that you reject the null hypothesis. Okay, reject the null hypothesis. So the grazing will cause the population of moth number to decrease. Okay, why? Because the grass could be the food for the moth. Okay, it could be the places to lay eggs. It could be places for the moth to hibernate. So if those places have decreased in number, so the population size will also decrease in number. Okay, so I'll put everything together now. What are the steps involved in doing the chi-square test? Is step number 1, you have to give your null hypothesis statement that there's no significant difference between O and E. That means O equals to E. Okay, then calculate the chi-square value, then calculate the degrees of freedom, then use the chi-square table or the critical, okay, right up, or critical chi-square value to find the property of your chi-square value, okay, and compare it with the property at 0.05. So if the property of chi-square value is more than 0.05, it will be at the acceptable zone. So you accept null hypothesis, that means O is equals to E. If the property is equals to or less than 0.05, so it's in the rejection zone, you reject null hypothesis, that means there's a significant difference between O and E. Then number 6, you do your conclusion statement. Okay, write your conclusion statement. So your conclusion statement is 95. So you have got, there is 95% confidence that there is or no significant difference, okay, between O and E. And please elaborate what is O and what is E. So this is your conclusion statement. So these are all the six steps involved in doing a chi-square test. I'm going to show you another thing here, which is also quite important, okay, in my earlier slide here, okay, this one. So what I want you to see here is, now, whenever we reject the final 5% data of the population, your conclusion is 95% confidence, okay? But if you reject at the final 1%, that means any chi-square value that are located at the final 1% of the population size in the chi-square distribution graph, right, reject at 1% level, then your confidence level of your conclusion is actually 99%, okay? Now, if you reject at a 0.1%, that means your final 0.1% at the end there, then you can conclude with 99.9% confidence, okay? For A levels, we choose 95% confidence. So we reject at the 5% level, right? Okay,